Everybody, so in this particular pre-lecture, we're going to talk about some biology review. So for some of you, this may be something that you've seen very recently, especially if you're an environmental major, but if you're civil, you may not have taken biology in a long time. So we're going to talk high level about some concepts that are important to this class that you're going to need to be aware of as we move forward. So many different types of microorganisms, small living things, are going to be really important when we talk about water and wastewater treatment and engineering. So they play a role in aerobic and anaerobic digestion in wastewater. So there's something that helps us remove things in that case. But occasionally microorganisms also need to be removed. So in the case of drinking water, we don't want what we call pathogenic organisms in our drinking water. So sometimes they're good and sometimes they're bad, depending upon the applications. So at a high level, microorganisms can be prokaryotes, which are small, simple, one-celled structures. They can be eukaryotes, which are larger cells, more complex structures. Each cell contains a membrane-bound nuclei. That includes things like fungi, algae, protozoa, or they could be viruses, which really don't fit into any category specifically. It's basically DNA or RNA in a protein coating. So that's kind of high level the types of microorganisms that you need to be aware of. So the microorganism that we're going to care about most of the time in water and wastewater is going to be bacteria. So single-cell bacteria are often classified by their shapes. They can be single cells, clusters, chains of cells. They can be rod-shaped, circular, all that type of stuff. So they have different shapes, and form basically fits function. They're different shapes because they have a different function based on their shape. So as an example, E. coli is rod-shaped and occur as single cells. So that information is really useful to know because E. coli is a pathogenic organism. So it's going to cause disease in humans. So if we want to remove it from drinking water, we need to know how it's shaped and that it occurs as single cells. So that's why that's important to us. Bacteria reproduce by what we call binary fission, and so under proper environmental conditions, they can produce very rapidly. So they have what we call an exponential growth curve. So they just continue to divide by themselves, and each one divides into subsequent divisions of organisms. And so sometimes we want that explosive growth, and sometimes we don't. So we just need to be aware of how fast those organisms are growing. And then finally, when bacterial cells die, their cells break apart or they lyse. And one reason that this is important to know is that sometimes bacteria contain toxins that we don't want. So we don't actually want to lyse cells in water treatment. So a good example of this is blue-green algae. They contain a toxin called microcystin, and microcystin is toxic, to humans and to animals, and we don't want to break apart the cells, so we need some way of treating to get rid of those cells without lysing them apart. So that's important to know as well. So a little bit more on bacteria. Most bacteria that we use in wastewater treatment come from soil, and very few are enteric. So enteric many times is going to mean that it's coming from within a specific organism. So instead we're trying not to use those bacteria, we're trying to use bacteria from soil. And the reason that we do that in particular with wastewater is that we want to focus on nitrifying bacteria. Nitrifying bacteria are going to convert ammonia to nitrate and nitrate, two different compounds. And many times they're autotrophic, so they can use CO2 for our respiration, and they can use it as a carbon source. So that's going to be useful in waters maybe with high alkalinity, where CO2 is going to matter in these specific waters. And then we can have different types of bacteria. We can have aerobes. Basically, these are microorganisms that are going to need oxygen when they are breaking down substrates and going through cellular respiration. So they're going to use oxygen molecules. And then we have what are called facultative anaerobes. These are kind of interesting because we use them in wastewater quite often. They can use oxygen in respiration if they want to, but they can also use different anaerobic pathways as well. So for those of you who maybe haven't taken biology in a while, you can think of as aerobics as you're doing cardio. You're going for a run. You absolutely need oxygen in order to do that activity. But anaerobic, you don't necessarily need oxygen quite as much or as in high volumes. So you can use anaerobic metabolism. So that might be something like weightlifting. And you can use both, right? So, or humans, we are facultative anaerobes. We're not microorganisms, but we have that facultative capability as well. So if that that analogy kind of helps you think about that, hopefully that's a good one for you. So if there's any one big concept that I want you to take away kind of from this biology review, it's really about growth phases. Not worrying so much about whether it's a prokaryote or a eukaryote or you understand cellular structures. I'm not worried so much about that. I think the important thing for water and wastewater treatment design is how bacteria grow. So microorganisms are primarily, their growth is related to the number of viable cells that are present and the amount of substrate or limiting nutrient that's present and other environmental factors. So on the next slide, I have all of these different phases defined out, but I think it's kind of nice to walk through this graph just so you have an idea of what's going on. So let me grab my laser pointer. So when bacteria start growing, we have kind of this like lag phase where the bacteria have just gotten into the specific area that they're in. And on this graph, time is on the x-axis, and then the amount of cells over time is on the y-axis. So this lag phase is basically where microorganisms have been introduced to a setting and they're not yet going through binary fission. If they're bacteria, they're not yet reproducing. And then we have what we call log growth. So over time, binary fission starts to happen. There's enough nutrient, there's enough substrate, so there's enough for the microorganisms to eat, basically, and they start reproducing, and it's exponential growth. This is a log growth right here. And then at some time, we have what we call declining growth. So basically what happens is there's so many microorganisms now, and there's not enough food for everybody. So the rate of binary fission and reproduction slows down. Then it's going to flatten off, and sometimes we call this a stationary phase. And then what's going to happen is we've consumed all of our substrate, we've consumed all of our nutrients, and there's too many cells. So we start to have cell death. So we have log death. Sometimes this is called endogenous decay, depending on what textbook you're looking at, what microorganism you're looking at. But basically, there's not enough food to go around, we've exhausted our limiting nutrient, and all the cells start to die. So if we think about this from a water and wastewater treatment standpoint, we care about this curve quite a lot when we talk about wastewater treatment. So we want cells to be growing and reproducing and consuming nutrients. We don't ever want to get to the point where we don't have enough nutrients and we kill off our cultures. So in wastewater treatment, we want to stay kind of in this log growth or stationary growth kind of area, because what that's going to allow us to do is always have microorganisms reproducing and taking in that substrate or that nutrient. And the nutrient or the substrate in wastewater treatment is going to be something like BOD or solids, basically things that we want to remove from wastewater. So that's why this is really important when we start to think about design. So again, just like the last slide, these are all of those different growth phases defined. This might be a great spot to pause the video and kind of write these down, write them in your own words, and make sure that you understand where they are. It might be good to try and use colors to define what these are and put them on, put those same colors on the graph on the previous slide, just so that you really understand what these different terms mean. So this slide is going to show you just a simplified version of those growth phases. So basically over time, we have time on the x-axis, we have number of viable cells on the y-axis, we have a log growth phase where everything is happy, we have enough nutrients, bacteria are dividing and reproducing, and then we have declining growth, basically where we start to use up that substrate and that nutrient, and then we have that endogenous decay phase where basically not enough nutrient anymore, not enough substrate. And we want to be in that phase, right, where that log growth phase may be just tipping into declining growth, but we don't ever want to get to the endogenous decay point in this particular graph when we're designing water wastewater treatment. So the other thing to briefly just be aware of when it comes to microorganisms is that many times they're classified by what they eat. So is their carbon source organic or inorganic? Do they use light? Do they use oxygen? Do they not use oxygen? So what you can see on this slide is just a couple different classifications that you may see, some words that may pop up. I'm not too concerned that you know these in very big detail, but these are some of the ways that microorganisms are going to eat and go through cellular respiration. So many times we care about those bottom three. We care about whether they're aerobic, anaerobic, or facultative anaerobes. So whether or not they use oxygen, they don't use oxygen, or maybe they could use oxygen in a specific case. On this slide, you're going to see just kind of the same as what you saw on the previous slide. These are different types of ways of classifying microorganisms. So by their energy source, is it from light or is it from chemical compounds? By their electron source, is it from organic or inorganic compounds? Their carbon source, and then the nutritional type is kind of their name. So again, no need to memorize these, but they may be words that you see come up in the future when you're designing water and wastewater treatment. And all they're related to is basically where do they get their energy and how do they use their energy. So now that we know basically how microbes grow, we need to know the different types of environmental factors that are going to basically affect that microbial activity. So in wastewater, we want to have microbial activity. In water, many times we don't. So we're going to need to know what factors are going to play a role in how those microbes are going to be reacting and moving within a system. So we need that biological treatment culture at optimum conditions, basically. So we need to choose treatments that are feasible based on water and wastewater entering the system. So in wastewater, we'll need a specific amount of biological activity to achieve adequate removals. In drinking water, we need to know whether or not disinfection will be effective in the system. So some quick environmental factors that affect microbial activity, some physical factors that you might see kind of pop up. Temperature is a big one. So this is going to double cell growth. Cells like it when it's warm, basically. And certain types of organisms are going to grow better at certain types of temperatures. Osmotic pressure is going to matter. So how much salt is in the environment and compared to water, so how much salt is dissolved in water is going to be important. The presence of molecular oxygen is important as well. So again, if you're aerobic or anaerobic, we need to know whether or not you need oxygen in order to function correctly. The presence of liquid water, right? So most organisms need to be in water to feed, like the organisms we're going to talk about at least. So they can't be kind of in dry, desiccated soil and be happy, right? They've got to kind of be in a liquid suspension. And then ultraviolet radiation. So ultraviolet radiation is at the lower range of UV. It's toxic to microbes, and that might be a good thing if we're trying to disinfect drinking water, but it might be a very bad thing if we're trying to have wastewater treatment. We don't want to kill off all of our microbes. Then there are also chemical factors that influence microbial activity. So pH is a big one. What is the alkalinity of the water? It's going to basically affect the rates of reaction. Presences of acids and bases, oxidizing and reducing agents, metals, ions, presence of specific chemicals, all of these are going to have an impact. So some of these are very toxic to specific cells, and sometimes we use that to our advantage, and sometimes that's a very big disadvantage. So basically what's on this slide, if you kind of look at these different examples, is all of the different ways that chemicals can impact your microorganisms. And again, sometimes we want to get rid of microorganisms, sometimes we don't. So knowing what chemicals are in the water is going to be really important. So over the semester, we're going to talk a lot about water quality and wastewater quality. And the reason that we're talking about all those different chemical factors is because it influences so much of what the microbial activity is within a water culture. And then finally, there's biological factors as well. So microorganisms don't exist in a vacuum. They exist kind of in an ecosystem with microorganisms. So there's going to be microorganisms eating each other and outcompeting each other within a culture. So you basically want to make sure that you have the quote-unquote correct microorganisms within your system to have the optimal function that you want to have. And then finally, temperature, right? So you're going to see kind of a growth curve here on the left that shows kind of an optimum temperature for a specific bacteria. But what you see on the right is there are different temperature ranges for different microorganisms. So each of these different colors is a different genus or different species of microorganisms. Some of them like it really warm, some of them don't, and that's important to know as well. The last thing that I'll mention here is that we call microorganisms basically that cause disease pathogens. So we have what are called waterborne communicable diseases. So basically a communicable disease is one that's spread from person to person. And unfortunately, or fortunately, I guess we have a really good example of this in our lifetime, which is COVID. Now, COVID is not waterborne, but it's communicable because it's spread between person to person. Waterborne diseases is basically a disease spread by drinking water. So there are many common waterborne diseases. We'll talk about these a little bit more. Typhoid fever is caused by a bacteria. Dysentery, there are actually two types of dysentery. One is caused by a bacteria and one is caused by a protozoa. Salmonella is caused by a bacteria. Giardia is caused by a protozoa called Giardiasis. Polio is a virus, hepatitis A is a virus, and Legionnaire's disease is a bacteria. And all of these communicable diseases are things that we watch out specifically for in drinking water treatment. And you'll see that they're different, right? There's bacteria, there's protozoa, there's viruses, and there's different treatments that we have to use for each of these. So understanding sometimes if you're dealing with a virus, a protozoa, or a bacteria is going to be important moving forward. And so to polish us off, I just kind of wanted to show this slide. It's kind of difficult to see these pictures, so I encourage you to kind of Google this yourself if you're interested. There are many waterborne pathogens out there, and those are the ones that we care about the most in drinking water. So when you hear these news stories about dirty drinking water causing disease, it's going to be one of these pathogens. And so there's many different types out there to be aware of. So to kind of sum all of this up that we've talked about, big things to take away from this lecture. In drinking water, we tend to want to remove organisms, but in wastewater, we want to feed them correctly so that they stay in the system. So there's different goals with each of these two. And then the other big thing to think about is that there are different environmental factors. There's physical, chemical, biological factors that are going to influence whether or not there's microbial activity in the system. And that's going to be really important when we think about designing reactors moving forward. So I apologize, this is a little bit of a longer video, but I hope that's helpful for you, especially if you haven't seen biology in a while, to kind of get a sense of why this biology is so important as we move forward with water and wastewater treatment.